since all algal mariculture depends on light, we shall start on light, then go through some of the principles of photosynthesis, and then get into certain methods of culturing algae and discuss the potential of it. Okay, so we start with the solar radiation and the way that light penetrates water bodies. So, as you can see, life is based on the planet, at least as far as we know, from the fraction of the solar radiation that is light, because other parts of the solar radiation cannot be used directly to support life. And as you see, the solar radiation comes from the sun's surface, that is at 5,760 degrees Kelvin, and it goes to the biosphere, captured only by plants, and then after making a few circles through the biosphere from plants to animals, and maybe from animals back that graze on plants, it supports all our biological actions, and then it is dispersed in space, in cold space, as heat. Now, the amount of radiation coming from a body is given by the Stefan Boltzmann constant that is of no concern. It is just used to convert the units of the solar flux into watts of accumulated energy. But the point that is very important is that the relation of the radiation of any body depends on the temperature of its surface, nothing to do with the material, nothing to do with the color, but to the fourth degree, which really is important to remember. It means that a very small change in the temperature of a body's surface, that includes of our own surface as a radiating body, any small change in the temperature results in a very large change in the radiation emitted from the body. And here you can see how we calculate the amount per 1 meter of the solar surface at the top. And then, of course, we have to multiply it by the size of the sun. Had the sun been colder, there would have been less radiation. And if the sun would have been smaller or larger, of course, also it would affect very strongly the radiation that is emitted by the sun and of the radiation that reaches us here. Now, another thing that is very, very important is that the peak of the emission of the radiation coming from any body shifts also not only in the total quantity, but also in its peak. And you can see a sample calculation for the sun, and it shows us that the peak of the solar radiation is in the green at 518 nanometers. And now if we look at the radiation emitted by bodies, and you can see on the black curves, that's the temperature, the surface temperature of the body, and under the area under the black line is the total, the integrated radiation, including the heat, the infrared, the light, and then to the left is the UV radiation. And you can see that with the temperature, the total area under the curve increases massively because, as I told before, it depends on the fourth degree of the temperature. And on the Y scale is, of course, the spectral units. Now, just to illustrate, you see in yellow the sun, but we have Antares, the reddish star that you see at the central of the Scorpion constellation, and it is colder, so it is redder than the sun, and you can see Spica that is much hotter, and there most of the radiation comes in the ultraviolet. And now, I don't want you to go into all the calculations, but if we look at the solar flux coming to the Earth, the amount of sunlight harvested or intercepted by Earth is really like the projected shadow that the planet, in our case the Earth, would cast on an imaginary scene behind it. But that is distributed over the surface, and the surface is 4 times larger than the shadow cast by any sphere. Now, about 40% of the sunlight reaching Earth is emitted back and not absorbed, and that is called the albedo. And you can very clearly see that the albedo of the clouds, of the ice, of the snow is close to 1. The albedo of the oceans is very low, so most of the radiation hitting the oceans is absorbed. You can also see the African plains, so the desert reflects a lot of the sun that reaches it, and you can see just to the right of it the belt of the tropical jungles of Africa, and they are dark, again, low albedo. And of course, if we want to calculate the amount of light energy of reaching any planet in the solar system, it really depends on the size of the planet. Of course, the larger it intercepts more of the solar flux, and as the distance increases, of course, you get much less sun. Now, all the planets, not all the planets, sorry, all the stars are receding from us, and that is called the red shift. And we know by what velocity and how fast they are receding, because we know the true position of the absorption of the hydrogen line. So if it is the extent to which it is shifted in the spectrum of a star to the right, we know how far it is moving from us, and that would have been true, of course, for any observer in the universe. Now, if you look at the spectrum of any star other than the sun, you see some image like that, and all the black lines tell us the identity of the different elements that are found on that star. On the sun, it's over 90%, it is hydrogen. Most of the remaining is helium, and the very few bits of oxygen, nitrogen, and carbon. Now, to remind you of the solar spectrum, and you have to remember that as we move to UVC, it is really deadly, and I shall say a few words about this later. Now, if we look at the solar radiation the way it hits planet Earth, you can see that most of the UVC, actually all of it, is stopped by the ozone in the higher parts of the atmosphere. Now, we got with the sun radiation, with the solar radiation, reaching the top of the atmosphere. Now, if we look, please look at the top, you see that to the left is the infrared, you have then the window of the visible light, and then you have the ultraviolet. Now, some of the gases in the atmosphere, you can see that methane absorbs very strongly in the infrared, which means that if the amount, the concentration of methane in the atmosphere increases, as it does because of the cattle, excuse me, of the farts of the cattle, that increases the greenhouse effect because it increases the absorption of thermal radiation coming back from the Earth. Then you see the nitrous oxide, and you see the ozone that is very, very important. You see that it absorbs 100% of the ultraviolet radiation. Then you see the carbon dioxide, you see water vapors, and at the bottom, you see the effect of all of them, because all of them exist in the atmosphere, but you can also see the window of the light, because very little, if any, is absorbed in the passage between the sun and the surface of the Earth after it it passes through the atmosphere. Now, you can see what would have been the solar radiation at the top of the atmosphere, and then you see, because of the absorption lines, so before you see what really reaches the surface of the planet. Now, okay, so we got to the surface of the water, and you can see that the UV, the UVC doesn't reach there, but still some ultraviolet is absorbed, reaches and is absorbed in the upper surface of the water body, and this is in Lake Superior, that is very transparent. If you had, as you see in a moment, if you have a suspended silt, or if you have lots of algae, of course the light will not penetrate that deep. But also, what is extremely important for the culture of algae, you can see that the red is absorbed very readily, and the blue is the most penetrating color. And you see the importance of it for the consideration of what algae we want to culture in a particular environment. And now you can see the relation of the light. You see the bars of the red that penetrates least, and the blue that penetrates most, and some of the remaining purple. And now you can see at the top the absorption lines of chlorophyll, and you can see what we experience every day, that that's why the plants are green, because chlorophyll cannot absorb the green. And it's very important, very interesting, because if you remember, we said a few minutes ago that the peak of the solar spectrum is in the green, and that part cannot be used by any green plant, land plants, and algae. But some algae, like the brown algae, have a trick. They have an accessory pigment that absorbs that green light that otherwise would have been wasted. And if we move further down, the area is that depth is dominated by red algae, and these red algae have pigments, again, accessory pigments that use the remaining spectrum of the light, getting at the deeper waters. And here you can see samples of the light penetrating Lake Kinneret. Pay attention, the bottom line is the color, actually, the wavelength, and you can see very clearly that at the surface we have the entire spectrum, and as we move further down, you can see that the red is absorbed very readily, and there is a major difference between these two situations. One is where you have a lot of algae, so you can see the absorption. And what happens is that in March, we have a massive bloom of algae, so you can see that the light is absorbed very readily. And please pay attention to the fact that the Y scale is log 10. So really the difference between the different depths is very, very steep. And again, you can see that at depth, the dominant color is the 560 that is not absorbed by the massive phytoplankton microalgae that multiply in spring in the lake. And here you can see the analysis of the same, and you can see that at the depth of 20 meters, almost nothing is left, and whatever is left is only some green light. But I want you to note that that change is mainly due to the amount of chlorophyll. You can see the bottom line shows the peak of the proliferation, of the multiplication of the phytoplankton algae, and you can see how it reduces dramatically the light penetrating into the lake. And this is very important to remember, that actually the most important factor in the attenuation of light in any water body are the pigments of the small microalgae. And really they are a little machine designed to catch as much light as possible. And again, many of the groups of the phytoplankton algae have those accessory pigments that allow them to use the otherwise wasted green component of the spectrum. Now, I'm sorry, you have in the box something in Hebrew. At the right, you have oligotrophic water with very little nutrients, with no algae, and you can see that the most penetrating component of the solar spectrum is the blue, and the red is absorbed readily, and the green also penetrates. Most of the absorption in that case is by the water itself. If we move to the left, it shows the situation, the opposite situation, where in the water you have lots of algae. You can see that now the most penetrating color is the green one. Now, the way light is absorbed in a water body is exponentially, and you can see at the top the radiation at a depth of z meters equals the intensity at the surface, multiplies by E, and depends on the number of meters we put. That's the Z in the exponent, and the KD is the absorption factor of the water with whatever exists in the water. Could be the water itself, the sum of the water itself, of any suspended inorganic particles, and primarily of the algae. And you can see an example how you calculate KD. It's very trivial, very simple. Now, remember that you know the KD, and now you can determine a very, very important factor that is a key factor also in the culture of algae. And that is the determination of the photic depth. And that is defined at the depth at which light from the surface is reduced to only 1% remaining. And why it is important? Because in very general terms, above it, there is more photosynthesis than respiration. Below it, there is not enough light, and so there is more respiration than photosynthesis, which means that you wouldn't like to create a situation in your culture that the algae, that there are so many algae that the bottom of the culture is in the dark, and you are really losing, you would be losing product. It's important to say that in the most transparent and oligotrophic water body in the world, that I believe is Crater Lake in Oregon, the 1% is at about 400 meters. By the way, 1% is still enough light for you to read in a book. But if we move to Lake Kinneret, for example, in fall, after the algal population used most of the nutrients and it collapses, the euphotic depth is about 20 meters. In spring, it reduces to 2 meters. And in your culture, for example, in the very oligotrophic and transparent coral reef region of the Gulf of Eilat, we move between 80 meters and 120 meters. And in a dense algal culture, the euphotic depth could be a few centimeters. And all of that are key factors in the design of your culture systems. So if you look at the profile, in this case, a marine profile, you can see that the red line represents the changes in photosynthesis with depth. Why doesn't it go further to the right at the top? Because light in the surface is too strong, and actually it inhibits photosynthesis. Then it goes down, as we said, as the light decreases, and we assume, and that's the yellow line, it is not exactly so, but it is good enough for our discussion, and that's the respiration loss. So you have to see the compensation depth is where the photosynthetic rate equals the respiration rate. But in order to have gains, you must understand to the bottom, the area under the right line has to exceed that of the respiration integrated over depth. And the critical depth is the line below which the total respiration exceeds that of the total depth integrated photosynthesis. Okay, and now it's a typical ecosystem. You can see that everything that happens there is supported by what happens in the upper parts, and those that are illuminated and can sustain the primary productivity, photosynthesis, most important of the microscopic algae that are really the meadows and the jungles of the marine environment. A brief introduction about photosynthesis. And as you know, it is the engine pushing it, supporting is the sunlight, and carbon dioxide is absorbed, and whether from air or in the case of algae, from the water, the dissolved carbon dioxide, CO2, and oxygen is released. We shall see in a moment further, and glucose is formed. And water is absorbed, in this case, by the roots. Now, it says glucose is formed, but in reality, the carbon chain of the glucose is then transformed into all the hundreds, thousands of organic compounds. They are all basically modifications of the carbon chains formed by plants in the photosynthetic process. Now, photosynthesis, the trivial equation of photosynthesis is carbon dioxide with water. You get glucose and you emit oxygen. Please pay attention that the arrow can be reversed, and then you see the respiration of everything, including all of us at this very moment. We take substrates, high energy containing organic molecules that really all originate in plants, and we burn it with oxygen, which we inhale, and you get the PG, which is the gross photosynthesis, minus respiration. You are left with what we call net photosynthesis. And really what we do, we cannot easily measure gross photosynthesis. We measure net, then we turn off the lights, we measure or at night in the natural environment, and we know the rate of respiration. We add it to the measured net photosynthesis, and we get the calculation of the gross photosynthesis. Of course, if we want to sell any algal product, the only thing we can sell is the net photosynthesis, because the respiration is deduced all the time at any moment during day and night. Okay, now a little bit about photosynthesis. We all were taught that what happens in photosynthesis is written at the top. And that is why we call it the glucose hydrocarbon, because it was assumed that CO2 is combined with water, and the oxygen in the carbon dioxide is the one that is emitted. The problem came with the advances of the availability of stable isotopes. Now, if we do the same, we take carbon dioxide where the oxygen is of the stable isotope of oxygen 18, and we turn the lights on, we discover to our surprise that the oxygen is not emitted. That's the oxygen in the glucose. And now, if we do the opposite, which means we label the oxygen in the water, we discover that all the emitted oxygen comes from water, from splitting water. But here we run into a new difficulty, because 6 molecules of water contain only 6 atoms of oxygen, and we have emitted 12 atoms of oxygen. So the real updated more formula of photosynthesis is written at the bottom line. We take CO2, 6 molecules of CO2, 12 molecules of water, and we have the oxygen and 6 new molecules of water. Okay, and now this was taken from a cruise, 2-month cruise in the Antarctic, and
sorry, Zvi, can you go back to the previous slide?
Sure.
So the problem is that the oxygen that is released by photosynthesis is part coming from. Can you repeat the last equation?
Yes. First of all, even the line before, it's clear to us that all of the oxygen that is emitted into the atmosphere from photosynthesis of the plants comes from the photolysis of water, which means the splitting of the very stable oxygen, very stable water molecule by the plants. But the equation is not balanced, because if you look at the equation, you emit 6 molecules of oxygen, but 6 molecules of oxygen have in them 12 atoms of oxygen. That means that to provide the 12 atoms of oxygen, you have to split 12 molecules of water. And that is the updated equation. By the way, all of you are invited to ask questions to stop me.
Yes, please, student, if you want to get in.
Yes.
So all the oxygen is coming from the water.
Yes, that is extremely important to understand. Now, here we show what happens at a date in the Antarctic where you have 12 hours of light, 12 hours of darkness. So we see during the light, the photosynthesis increases, and with it, the biomass in the algae. By the way, the line is not straight, it's exponential, because during that time, the algae also increase in numbers. Otherwise, it would have been a straight line. The dashed line above it is what would have happened if there would have been no respiration. So the 2 lines on the left show you the net, that's the black, and the gross rates of photosynthesis. Now you see that during night, there is only respiration, and actually the algae use up some of the stuff that they accumulated during the day. Now the difference, you see, the P1 plus D is what they gained during such a period of 24 hours. But imagine now that you had, for example, only 6 hours of light. Then, can you see the arrow? Can you see the arrow?
Yes.
Stefan?
Yes.
The photosynthesis would have stopped here, and then respiration would have been going like this, which means we would have ended up with less biomass than we started. What happens during the long summer? Photosynthesis goes on, and the night is short, so there is very little loss. And that really explains the function of the southern and actually northern latitudes. because what happens, the days get longer, and there is an extremely fast increase in the multiplication rate of the algae. And in reality, in winter, the losses are reduced. Why? Because of the low temperature, respiration decreases significantly. And that is another just a comment. Respiration has a very strong factor effect of temperature. At photosynthesis, very little. Let's say photochemical processes are very, very weakly, if at all, influenced by temperature. Okay, how do we measure fluorescence? So first of all, one way is we label the carbon in the CO2, and either it's in the air with the land plants, or we add it as a salt, as a sodium carbonate or something where the carbon is labeled, and then
Zvi, I have a question. So with increasing temperature, let's say the photosynthetic system is more or less stable, but it decreased the respiration.
yes,
It's increased the respiration. With temperature, the respiration increase.
yes, but what is important is actually the phytoplankton in the Antarctic and Arctic survives the months of darkness only because there is such low temperature, because the respiration losses are minimal. Otherwise, there would have been no algae left to start the cycle in spring. Is that clear?
Yes.
Okay. So I would say that in aquatic experiments, what we do, we add some labeled carbon, and we look for it in the biomass, actually in the algal biomass, and from that we know how much carbon-14 was absorbed. Now, we are not interested in the carbon-14, we are interested in the carbon-12. But we know how much carbon-14 we added related to the ambient carbon-12, so we know how much photosynthesis is going on. We can also measure the rate of emission of oxygen and calculate the rate of photosynthesis from the change in oxygen concentration. That we do only in water, because in the air the amount of oxygen is so high, 18% or so, that we shall see very little change. But in the water we see a massive increase. We can also measure it from fluorescence and from photoacoustics. I don't want to get into that. Just remember that there are additional methods to do, and we estimate, as I said, the gross photosynthesis from respiration photosynthesis. Now, if we imagine any photosynthetic apparatus, so we get sunlight, a large fraction that can be anything between, let's say, 30% to 100% is immediately converted to heat, which means that a variable fraction of the absorbed light by any photosynthetic apparatus, a large fraction is lost. And the healthier the system is, that fraction goes down to around 40%. If the system is not doing well, let's say a terrestrial plant that didn't get enough water, the losses will be massive, and they can really consume everything, and we are not talking about respiration. And then you have the partitioning between photosystem 1 and photosystem 2. You have very small emission of 2 to 5% as fluorescence. And you see the green arrow, that what is left for really for the photosynthetic activity, but in terms really of the basic molecular absorption processes of light in the photosynthetic mechanism. Now, I want to show you how we basically measure photosynthesis here in that case, in the aquatic environment. We take whatever organism, it can be an alga, it can be a suspension of microscopic algae, and we expose them to different amounts of light by different amounts of the screen or controls with no screen at all. We leave it in the water for so many hours, then we filter it and we look how much carbon-14 was absorbed at each light intensity, and from that we can create a profile as we saw. In this particular study, we were looking at corals. I should say a few words about this later. Here you can see a machine, submersible respirometer, where there are electrodes in each chamber. You have 3 replicates, and here we measure photosynthetic rates or respiration rates by the changes in oxygen in any of these chambers. Here we see the major method to study in the marine system, in the marine environment at different depths, to study the rates of photosynthesis. The system is called the rosette, and it's really cylinders. We submerge them and we close them from a computer on board of the ship, so each of them at the different depths. So we have a series of samples of water with the algae at different depths, and the next thing, we add to all of them the carbon-14. We incubate them at different light intensities measured at the different depths and at the different water temperatures to which they are exposed during the Antarctic year. And here you can see the filtration so that only the algae are left, and we look at how much carbon-14 was absorbed, and from that we study the photosynthesis rates at all depths, and we can integrate them and know how much was the total photosynthesis. Here you can see a very important thing. You can see at the top the different rates of photosynthesis, primary productivity photosynthesis on land, and you can really see that it is influenced only by one factor, and it is the amount of rainfall. At the bottom, you see it's parallel in the aquatic system, and what I marked in purple, in pink, are the areas where there is very little photosynthesis. So photosynthesis on land is limited by water. By what is photosynthesis limited in the sea? It's limited by the availability of nutrients, primarily nitrogen and phosphorus. And you can see that reaches the aquatic systems from the land. It is washed and comes down after rain, and you can see really also the difference in South America between the West and the East, and also in Africa, and that is because the rotation of the end of the planet brings to the surface in a process called upwelling nutrients from deep water. Now we are moving further. You can see that I'm comparing.
Sorry.
Yes.
If you go back to the previous
Sure.
slide. Okay, for sure, the photosynthesis depend on nutrients coming from land.
Yes.
But if you look at the open ocean, let's say in the middle of the Atlantic,
Yes.
you see there is also another maybe factor depending on temperature.
Maybe I have to add a few words of explanation. You don't see this pink in the far south or far north. And really, the oligotrophic, I mentioned it, with the areas with very low nutrient contents in the warm waters of the tropics, let's say latitude 30 up and 30 down from the equator, it's because you have thermal stratification of the water column. So there is light, warm water floating on top of cold water, and there is no mixing, so that nutrients that are released from dying organisms, from dying algae, remain sealed in the cold, deep waters, dark waters at 2, 3, 4 kilometers below the surface. So there is no, there are no available nutrients in the euphotic zone, and that is why these are so poor. And actually, if you think of it, the fisheries, the world fisheries, are in the gray areas where there are enough nutrients to support phytoplankton, and that these are eaten by fish and crustaceans, and they are eaten by larger fish, by carnivorous fish, and these are the fishing grounds of the planet.
Yes. Yes.
I want to go through one more slide, and then I suggest that we break up for a few minutes. I'm sure that everybody needs a coffee now. And just look at the upper. Okay, first let's look at the columns. The left column is how many kilometers, square kilometers, are covered by each of these units. You can see the terrestrial ones at the top, the marine ones below. And then we see at the range of the primary productivity per year. And then you can see the next column is the mean. Then you multiply that mean by the area, and you can see that system, that biome, how much primary productivity it contributes. Of course, primary productivity is only the net, because what is respired is lost. Now you can see the amount of biomass, and you see the range, and in that case, it's kilograms per square meter, which equals tons per square kilometer. And then you can see the global contribution. But I want you to look only at the following: at the mean in the tropical rainforest, which is, you can see it, it's in grams per square meter, which equals to tons per square kilometer again, but you can see that you have per year 2,200 grams per square meter per year. Now, if you look at the next column of the mean of the biomass, you see actually that 45,000 grams, 45 kilograms produce in a year about 2 kilograms. Keep that in mind. 45 kilograms produce about 2 kilograms a year. Or in another word, it would take many years, 20 years, to produce another 45 kilograms at that rate of production. Now look at the bottom, at the upwelling zones, the most fertile zones in the ocean, and you can see that the average productivity is 0.5 kilogram. And here I have to lower this, and this 0.5 kilogram is produced by 20 grams. So the 20 grams at that rate are doubled every few weeks. And that really reminds you, shows you that the productivity per unit biomass of the phytoplankton is much, much higher than that of the jungle trees. And now think of a tree. What fraction of its biomass really does the photosynthetic work? Only the leaves, not the branches, not the main bulk, the trunk of the tree, not the roots, not the flowers, not the fruits. So all of these are basically parasitic on the photosynthetic process. Now, furthermore, in a tree, in a terrestrial plant, you need transportation systems to bring water from underground to the leaves. You need another system to convey sugars from the leaves all the way to the flowers, all the way to the roots. You have no such expenditures with the algae because you have the water and the nutrients surrounding them. There is an additional consideration. You know, the plant biomass is thousands of times heavier than the surrounding air. So you need mechanical structures to support the tree, to support. All of these are wastes in terms of the photosynthetic products. Really the density and the weight of the algae is more or less like that of the surrounding water. You don't need that. You don't need flowers. You don't need roots. So from here, you begin understanding that basically the production by algae must exceed by a lot that of corn or of any terrestrial plant. I'm trying all the time to divert your thinking towards the mariculture of algae. Now, if you calculate it, what I said before, you can see that to double the third line, to double the biomass of the trees will take about 25 years. And here with the phytoplankton, it will take 1/25 of a year. And the ratio is that per unit biomass, the algae are about 500 times more efficient than terrestrial plants. Let us break up for a few minutes.
Okay. Okay. Let's take like until 15, like a 10 minutes, no, 5 minutes, 10 minutes. How much you need to see?
Yeah.
Okay, 10:15. 10:15 in Italy, 11:15 in Israel. Okay, we will start.
Perfect. Yeah.
Okay. Thank you.
Okay.
Stefano?
Zvi, you need something?
Yes, tell me, Fiorella, am I going too fast?
No. No, no, no. Everything is very, very clear. I think probably the questions will arrive at the end of the presentation from everyone.
I'm here. I was in the other room. No, everything is okay, and maybe this okay. This black rectangle here that is on the desk.
Yeah.
Can be turned in his camera, I guess so. But
my camera is open.
Your camera is open. Yes, we see you.
Yes, but I don't see myself. Doesn't matter.
Doesn't matter. No, it's okay.
It was okay before, so we can continue that way.
Ciao, ciao.
Okay. And Stefano, Fiorella told me that I'm not going too fast. It's okay?
Wait, Zvi, I think he's on the phone.
Stefano?
He's on the phone, Zvi.
No, okay.
Are you teaching in this, in these days, in this current time, Zvi, or you're not?
I begin teaching next week.
Next week.
Yes, but that is for the program in Netanya Academic College, not at Bar-Ilan.
Oh.
And that is Masters in Business Administration on managing the resources, the environment, of energy, and of water.
Wow.
And most of the people, it's an MBA, and this is mostly for engineers working in electricity producing plants and similar water systems. And these were trained as electrical engineers. They never heard about photosynthesis or about plants.
Yeah.
And they learn in the course how they can destroy them and harm them.
How they can get energy from them.
No, and it's really important because the course really changes their attitudes.
Is this like a course with a biological background that they follow?
No, they are required to take in the summer a preliminary short course on chemistry and biology. And that I don't teach. When I come, they are meant to be already after that course.
Do they interact with you? They ask questions, they look interested?
Yes, very much so. And we also discuss in detail all the options of sustainable energy systems.
Mm-hmm. Yeah, it's a very up-to-date topic.
Yes, and actually it's a very different experience because all these people are, of course, much older because and they get 2 days off work to take that course from their different employers.
Oh, so it's the actual employer that want them to follow this course.
Yes, actually they are paid for it. The employer pays for the course to the university, but it also increases their salary.
Really? Wow. Nice. And you have many students in this lecture, in this class?
It changes a bit between years, but between 50 and 60.
Mm.
And the Bar-Ilan course is open to, mostly taken by third year BSc students. And this year it was very popular. We got 90.
How many hours do you have to teach?
At Bar-Ilan, it's a 2-hour course for 1 semester. And at Netanya, it's 3 hours a week for 1 semester. It's the first semester I teach at Bar-Ilan, the second there. And at Netanya, they also have to, in a second course that I teach, it's a seminar course, and they have to prepare a seminar. And in the years when there is no corona, Bar-Ilan, they also prepare, and they get a bonus for it. They also prepare posters, and we have a very beautiful poster exhibition at the end of the course.
Oh, nice. So they haven't tried to shift this into an online exhibition, no?
No.
It's difficult.
It's not difficult, but I didn't find it important. The only thing I required from them was a 5-minute PowerPoint presentation, and that worked very, very well. And they were encouraged to pick as original topics as possible. And in the Natalia course, where they need to make an hour presentation, also there were some topics that were amazing that I never thought of. For example,
Zvi,
yeah.
I leave the room 1 second to go to pick up a coffee at the machine and come back. Okay, then we start. 1 minute.
Yeah, okay. You tell me when to start.
Okay, thank you.
Fiorella,
yes.
So one student prepared a seminar on the ecological meaning of the different burial methods of the dead.
What?
If you bury in ground, if you cremate them, if you feed them to the birds, like in the Persian culture, and so forth.
Wow. The ecological meaning.
Yeah, so it was really, really so interesting. And another girl prepared on the different ecological disasters in movies and literature.
That's a nice one too, yes.
And then ecology in the Bible. So it was, and others made the presentations on kind of the things that you would have thought immediately. Ecological effects of electricity, high power lines, of cell phones.
Less original, a bit less original.
Yes, and the use of gray water. You know what is gray water?
Yes. So the ecological effects of
No, how you recycle, you know, you have to plan when you build a house or an environment to separate black water from gray water.
gray
You
water.
know the difference between them?
No,
Black water,
actually I don't.
black water is the toilet.
Yes.
Gray water is from your laundry and dishwater.
Ah, okay, no. Okay.
Actually, Israel is #1 in recycling wastewaters. 80% are recycled.
Wow. Yeah, Israel is
Yes,
it's powerful.
but in exchange, our prime minister is not recycled.
Not everything is recycled, huh? So you still enjoy teaching a lot, Zvi?
I teach one course every semester, and actually I've been doing that for many years, more or less. And we now have in the lab actually three grants.
3 grants.
Yes, small Israeli. One is on the light and climate in a sea cave we have in the north of the country, where the light climate is very complicated, change with the hour, change with the season, and we are studying how the algae accommodate, manage with that by changing the composition of the community, changing pigmentation. That's one project. Another project that David has is a study by reflectance spectrum, the origins of pigment in tissues found in archaeological digs, excavations. And it's very important because you find what plants and how were they made, and you can even say, okay, these plants were not grown here, so it shows that there was commerce coming from one place. It's very, very interesting. And the third one, we have a project funded by a company for using microalgae as fertilizer.
Okay, Zvi, I think we can restart.
Good. Okay.
You want?
Okay. All right. So we look now. Can I start?
Yes, please.
Okay. We look now at the ancient ocean. Now, iron is one of the most common elements on our planet. So the ancient ocean was saturated with the soluble Dubinsky oxide of iron, which is water soluble. Now, about 3 million years ago, you already have blue-green algae, cyanobacteria, and they emit oxygen in their photosynthesis. Now, that oxygen immediately combines with the iron and to form the trivalent iron oxide that differ from the two-valent is not soluble, so it sinks immediately. Now, for about almost 2 million years, all the oxygen created by the photosynthesis of these organisms sinks to the ocean floor. So for that very long period, there is no oxygen coming into the atmosphere. No oxygen means no ozone. No ozone means that UVC reaches every surface, which means there is no possibility for land life. In the water, it is possible because after 20 meters, 30 meters, no UVC is left. So during that period, life expands and biodiversity increases in the sea, in the ocean alone. But what happens at the beginning, you see the first 1.5 million of years after the solidification of Earth and the formation of the oceans, there is no photosynthesis. Then there is photosynthesis, but all the oxygen is absorbed by the iron oxide and sinks to the bottom. Now, about 2 million years ago, you begin the release of oxygen into the atmosphere, but nothing is left because that oxygen oxidizes all the material on land that never saw oxygen before. And then 650 million years ago, all that has been oxidized, and very rapidly oxygen in the atmosphere reaches our present levels. And that is the time when life can leave the oceans and populate land. And that includes plants and animals that could not get on land before there was the ozone shield making it possible. All right, just to remind you from the solar energy, how is the partition between land and oceans. Now, this has been revised, and now we believe that much more of the photosynthetic process accumulation takes place in the oceans, about 50-50 with land. And the reason is very peculiar because in the early years, much of the very, very tiny algae that populate the oligotrophic parts of the oceans just went through the filters and were not accounted for. And now you can see the difference between how much the very little fraction that goes into agriculture and something that we usually don't think of. A lot of agriculture is not supported by sun. If you think of the tractor, the metal, the fuel, all comes from fossil reserves formed finally by photosynthesis, but millions of years ago. So this is a kind of a thought that is not always taken into consideration. Okay, now plants and algae photoacclimate to light levels. Again, you'll be able to place it later into your thinking of possibilities for algal culture in the world. Okay, just one more comment. I use the term acclimation different from adaptation. And usually in the language, people don't take care of thinking of the difference. Now, adaptation means change in the genome, takes many generations, can take many, many years. In bacteria can take maybe years, as we see now with the coronavirus. But with higher organisms like people and elephants, it takes thousands of years. Now that differs. Adaptation differs from acclimation. Acclimation is with the latitude of phenotypic change without change to the genome. Now, what you see is an algal cell on the left, grown under very high light, on the right, under low light. And you can see the change in the black lines are the thylakoid membranes, those that carry the photosynthetic pigments. You also see the little white areas, droplets of beta-carotene. And that serves under high light, absorbs part of the light, and shields that way from the delicate photosynthetic apparatus from photoinhibition, from damage by excessive light. You can also understand that on the left, not all the photons are absorbed. On the right, the opposite is true. You don't need the photoprotective pigment. On the other hand, any photon hitting the cell will be absorbed by one of the layers. By the way, that alga is very interesting already in the context of algal culture. It's Dunaliella salina that grows periodically in the Dead Sea if there is a rare rain event. Then the fresh water flows on the very heavy brines and gives the opportunity for that alga, where there always is an inoculum in the surrounding waters, to proliferate. Now, the high salt content in the water takes the water out of the algal cell by plasmolysis and reduces the tension in the cell membrane. And that starts a biosynthetic cascade producing glycerol. Now, glycerol is a small molecule, so it's osmotically active. So it draws water back into the cell. The tension to the membrane, to the cell membrane, is reestablished and the process stops. Now, that was discovered by a team at the Weizmann Institute, and they issued a patent for the production of glycerol by algae. It was very successful, except one problem, that was about 20 times more expensive than the glycerol traditionally made. So that was abandoned, but luckily they discovered that under high light and nutrient limitation, that alga produces vast amounts of beta-carotene. Again, think now about algal culture. Per unit weight in Dunaliella, there is 50 times more beta-carotene than in carrot. So that created, and you'll see an aerial photograph, a system for culturing the alga in Eilat and in Western Australia. It's very valuable. for as a dye in food, but also in human diets as a food supplement, and it is an antioxidant and has other functions.
So they actually succeed to produce beta carotene by Dunalia salina an industrial scale. I mean,
yes, it is a very successful operation. It's interesting. It was developed by Israeli company and they were inefficient. They were losing money, so they sold it to a Japanese company that was very successful, and they make a lot of money, and they sell it as Jerusalem Dunaliela. By the way, the alga doesn't have a cell wall, so you don't have to extract. You can just put it in pills and give it to people, and they are very happy to pay money for it. And now I want to show another case of extreme photoacclimation. What you see now are polyps. I'm sure that Stefano will be very happy to meet these friends of his. You can see the colored spots. These are microscopic algae, 10 microns diameter, that lives inside the coral polyps. The polyp is quite transparent, so the algae can get as much light as they need. And remember that corals succeed in tropical waters where there are no nutrients. So inside the polyp, you have, from the excretion of the animal host, you have sources of nitrogen and phosphorus, and from the host, the animal respiration, they get CO2 that is essential for photosynthesis. Okay, now, all kinds of corals have these symbionts that were termed over 100 years ago as zooxanthellae, which means yellowish cells within animals. And here I took that photograph of a coral harboring about 1 million of these algal symbionts per square centimeter of its living tissue, covering the calcium carbonate, the aragonite skeleton. And the left one I took from very low light, 70 meters deep, and the right one from the surface. Now, first of all, it makes sense. We understand that the energy, 95% of the energy requirements of the coral system is provided by photosynthesis of the algae, and that is a unique system. It would equal growing us wheat inside our bodies, or rice, or whatever, or potatoes. Okay, now it makes perfect sense that under low light, you would be as dark as possible to capture as much light as possible. And the opposite is that the intense radiation in the shallow waters of coral reefs. So you would like to catch the minimum light needed to sustain the needs of the algae and the host, and the remainder to penetrate and be reflected by the white skeleton. And really, what you see makes sense. HL is highlight, LL is low light, LL divided by HL. And you can see that there is about 4 times more chlorophyll per centimeter square under low light, see at the bottom line. But when we counted the cell numbers are not different, because when we looked at the almost black coral, we saw that there are 2 possibilities. That the animal facilitates to have more algae to compensate for the low light, or that the numbers do not change, but the pigmentation in the cells change. Now you can easily see that in each cell there are about 4 times more pigment in the low light zooxanthellae than in the high light. So the difference per area in this case is totally explained by the different acclimation of the algae. And just to give you an idea how a cell of the low light zooxanthella looks, it's all packed. You understand it's a cross section, so it's all packed with these photosynthetic pigments, and of course they carry the excess, the high amount of pigment of chlorophyll needed. And if you see at the same section, at a similar section of the highlight coral zooxanthellae, you can see that there is much less pigment, and you see also the droplets of the photoprotective carotenoid. And really that is the model for high and low light acclimation in general. On the left you see low light, you see many thylakoid membranes, a lot of pigment, virtually all the light is absorbed. And actually in the dark coral, over 95% of incident light are absorbed. In the case of the highlight coral, about 5% only. So you see less membranes, less thylakoids, and less pigments, but that is nothing to do with corals. That is true for any algal cell. Now, if you look to the photosynthesis to radiation to light intensity curve, it is called the PI curve. Today, for some reason, the I has been replaced by E instead of irradiance. Its energy doesn't really matter. Now, look please at the two graphs. One is labeled HL, highlight, and one is labeled low light. So look at the dark, at 0 light intensity. You see that the oxygen doesn't increase, it decreases because it's used by respiration. But you can see that the respiration rate of the high light algae is much higher than that of the low light. Why? Because they grow, they multiply. All of that requires energy. Okay, then you see that the crossover of the 0 line, that if you remember, it's the light intensity at which the photosynthetic rate equals that of the respiration requirements. And then you see that the low light line is steeper. I'll get to that. The angle at the bottom, the alpha L against the alpha H are different. We shall get to this in a moment. And you see that the low light algae are saturated at a much higher, at a much lower light intensity, and the high light algae are saturated at a much higher light intensity, which means that when the line becomes horizontal, that at that point, when you add light, you do not add any additional photosynthesis. At that point, the photosynthetic system works as fast as it can. And there is much less of the system in the low light acclimated algae, because there would be no need to make more of that energy costly system when in real life, not in the laboratory, you never meet high light. Okay, now please look at the slope. that slope, the tangent, you remember your trigonometry lessons many years ago? What is the tangent?
I have a question. Why the highlight system become saturated highlight system becomes a much higher light in irradiance?
Because in each of these algae, there is much more, for example, of the enzyme rubisco, that is the key enzyme in the assimilation of the carbon dioxide. Mm. In the other case, in the low light algae, there is no need for that. And we measure it. There is a dramatic difference because they wouldn't make that enzyme when there is more than needed if there is low light, because you don't need much of that enzyme to process carbon dioxide in photosynthesis. Okay? Mm-hmm. All right, so look at the angle. The tangent really is photosynthesis divided by light intensity. So the steeper the slope, higher the intensity, higher the efficiency. The angle of that line is the efficiency of photosynthesis, what is called the quantum yield of photosynthesis. That is the product divided by the light. And of course, that translates in our algal culture system into how much biomass or product you make divided by the amount of sunlight, and the amount of sunlight is proportional to the area. So it really translates, equals to euro per square kilometer. So that very basic photophysical or whatever factor translates into direct economical calculations. And here you can see in algae growing at the bottom at different light intensities, and look only at the line with the black dots. You can see that at the lowest light intensity, and look now at the extreme right, you need only 8 photons to assimilate 1 molecule of CO2, or 8 photons to emit 1 molecule of oxygen. And it is very simple. You need 4 electrons from water to oxygen, and these are emitted in 2 photoprocesses, photosystem 2 and photosystem 1. It doesn't matter. It's not a course in photobiology. But you need at least, you cannot do it with less, 8 photons. And you really see that algae acclimated to low light work at the absolute physical highest efficiency possible. Now, if look at algae grown at high intensity, at 1000, doesn't matter the units, Einstein, that's quanta per square centimeter per second, doesn't really matter. You need for the same to emit 1 molecule of oxygen, you need something like 100, which is 10 times less efficient, 12 times less efficient. So now let's go back. If you think at an alga with a lot of pigment and against an alga with very little pigment, and they are exposed to the same light, so you see that the to the ambient light, you see that the light can be 100 times higher in the high light grown algae than in the low light culture. So the difference in light input is 100-fold, but the darker the pigment-loaded algae get 4 to 5 times more of the photons. So the 100-fold difference is now reduced to a 20-fold difference. And then if you look at the efficiency, the quantum yield efficiency, it is reduced about 5 to 10 times more. So now the difference of 100-fold intensity in light is reduced to a 5 times efficiency in photosynthesis. So you have two phases. One is really optical. They are just intercept more light. And the second one is kind of biological, photochemical. So here you have two components of the photoacclimation process. And here you have a Petri dish with seeded with Haematococcus, and Haematococcus translates really into the red sphere. And that alga is a green alga, is a chlorophyte of fresh waters. And you can see it was grown against a light gradient. And you can see at the top it's dark, so algae cannot grow. Then you get to the green area, and you see that under high light, these green algae save themselves by making a lot of a red pigment that is a chloroprotective pigment called astaxanthin. To give you an idea, astaxanthin sells at thousands of dollars per kilogram. Now, Carol.
Excuse me.
Yes.
Can you go to the previous slide, not the other one? I didn't understand properly how you can explain from this slide why there are two components, what there is like a physical factor and a biological factor.
Okay, the physical factor is really explained here. You harvest more light, you harvest less light. And in the case of the coral, for example, or in the case of Dunaliella, you have 10 times more light harvesting chlorophyll in the low light adapted acclimated cells than in the high light ones. Okay, so that is the optical component. Is that clear? If you are darker, you absorb more light. Okay, but here you have a different efficiency. Once the light is absorbed, a higher efficiency of using the light that you absorb. And the efficiency is 8, in this case, let's say 10 to 100, which is a lot of difference. So these two add up to the effectiveness, to the incredible effectiveness of the photoacclimation process. And here you can see the function of the photoprotective astaxanthin. Now, astaxanthin has a commercial value because you have to include it in the food pellets of salmon. Without it, the salmon meat will be white and will have no commercial value. Okay. Also, it has a physiological role, but that's another thing. But now you have two groups of carotenoids with very similar chemical structure. The light harvesting are fucoxanthin and peridinin. Fucoxanthin is the one in the brown seaweeds, and peridinin is in diatoms and in the zooxanthellae. So that is the pigment that harvests the green light and transfers with no loss to the photosynthetic apparatus that cannot harvest the green component, the abundant green component in the solar flux. And then, so these two pigments increase along with chlorophyll when plants are exposed to low light. That increases the fraction of light absorbed. Then, under high light, you have beta-carotene in astaxanthin. Both of them, what I call our sunglasses of the photosynthetic apparatus. And also the xanthophyll cycle pigments. I didn't want to go into this. And therefore, there is no, in such studies, there is no sense at all to measure total carotenoid, because part of it they have opposite functions. One increases the light harvesting and the other reduces the light harvested. Okay, now we're beginning to go into practicals. If we look at the growth, irradiance under which algae are grown, you see that certain things decrease as light intensity increases, chlorophyll A decreases, polyunsaturated fatty acids that are very important as food additives, and antioxidants, because they reduce. Thylakoid area reduces, the quantum yield decreases. And the opposite is true for the light harvesting pigments like beta-carotene, the light protective pigments like beta-carotene and astaxanthin. They increase when you expose the algae to high light. And of course, the doubling rate and the maximal photosynthetic rate, but also respiration increases. Now, when you start making algal ponds in the sea or in fresh water or whatever, you can play, because first of all, you can choose the latitude on the planet, if you have more light or less light. Of course. Okay.
Yeah, sorry, it said also the depth of the culture is.
Wait. Then you can decide where to construct the system if there is a lot of cloud or less cloud. And then, as Stefano said, you use the depth of the pond. So to the left, you get low light acclimated cultures. To the right, you get high light acclimated cultures when you combine all of these factors. You can also change the concentration of the algae. So the higher the concentration, there will be less light absorbed to each one of the cells, because part of the cells find themselves in the shadow of other cells. So here you start thinking, you want to make money, you start thinking where you grow the algae, where you set up your systems, what depth you make them, and what concentration you allow the culture to reach before you harvest. Now, you know that you have an increase in temperature of both oceans and land. I don't want to go into this, is the greenhouse effect, and just something that is very exciting, and you'll see in a moment why it is important. This team is the Russian team on the Vostok station on Antarctic. And they went to the highest elevation of the ice, the depth of the ice at the peak is 3 kilometers, and they drilled without melting the ice. So that these 3 kilometers were accumulated in the course of 400,000 years. You have snow coming, next year snow on top, and you know why snow is so light? Because most of it is air. So when the snow is condensed, compressed into ice, in that ice are captured samples of the atmosphere at that year. Now you can see that you can count the years, summer, winter. So you know the exact age of each one of these samples. And they are kept, the 3 kilometer cylinders are kept for study. Now, if you take a sliver and melt it, you get back the atmosphere, and you can measure the amount of carbon dioxide in each of these 400,000 years. Actually, they found now a deeper place, and it's 600. Why not more? Because under the intense pressure of these 3 kilometers, at the bottom, the ice flows like plastic. So it's accumulated at the top and escapes at the bottom. So really, you have 400,000. Now, from other measurements, and I don't want to get into that, you know exactly to 1/10 of a degree what was the temperature at that time. Now you plot the temperature against the CO2 concentration, and you see very clearly that the greenhouse warming effect is created by changes in the CO2 content of the atmosphere. And if you look now at this tracing of the CO2 in the atmosphere, you see that it increases very rapidly and exponentially, not linearly. And we are talking in recent times. By the way, why do you see the zigzag? By the way, that was taken in the Mauna Loa Observatory on the big island of Hawaii. And that was chosen because it is very far from any industrial pollution. You see the CO2 goes up and down, and the cycle is 1 year. Why? Because during the growth season, the plants absorb CO2 and reduce the content of CO2 in the atmosphere. In fall and winter, leaves fall, there is less photosynthesis, and the leaves are decomposed by microbes and insects, and in their respiration, they emit CO2. So you can see that the biology really plays a role in that. And also the increase is due to the increase in human population, in cow herds, and in the amount of automobiles that we all use. Now you can see that for 1,000 years, the CO2 content in the atmosphere was below 300 parts per million. Then we reached the Industrial Revolution. That's the blue section. And these are different of the ICCP Institute for the prediction of climate change. These are different scenarios, and steepest is called in that in our literature as business as usual, which means our behavior regarding consumption of meat and growing the animals for meat and the consumption of fossil energy, if it continues as is, which really was the view of the last president of the United States, Donald Trump, and the lowest is if all the measures dictated by the Paris Protocol are taken, still the CO2 will continue to increase, but will level at just twice its present content. Okay, but as that increases, you see on the left, that's the map of the Hawaii archipelagos of where that was measured. Now, as the atmospheric CO2 increases, so does its concentration in the seawaters. That's the purple line. But that causes a reduction in the pH. So we have the increase in temperature, and we have the acidification process of the oceans. And you can see that for 25 million years, the pH of ocean was more or less constant, and then it dips very, very fastly during the current industrial revolution. And you can see the changes in pH, and you can see that the main acidification takes place, unfortunately, where corals abound. And as you acidify seawater, the process of deposition of calcium carbonate shifts from its deposition into its solution. And that is an experiment by two colleagues, Danny Chernov and Maoz Fine. And they took a coral that's the upper left. They exposed it to acidified seawater, and the skeleton, the calcium carbonate dissolved, and you see that the polyps changed into small anemones. Then they raised the pH back, and you can see that they begin calcifying again. Now, this is very relevant to our talk, because all the shellfish industry depends on the calcification. Snails and crustaceans and sea urchins and clams and oysters, all of them are adversely affected by...
Yes, 1 second, Michele. Okay.
Okay.
Yes, are affected negatively by the acidification of the oceans. But photosynthesis goes the opposite way.
Okay, Zvi, before going to this slide, there's Michele as a question.
Yes.
Yes, it's just a curiosity. In the previous slide, I see that in Mediterranean there's no data. The previous one.
Yes.
And seems that in the Strait of Sicily there's a basic pH.
Yes. Yes. It is not detailed. I didn't find a similar map of the Mediterranean, but as Stefano can tell you, there is an ongoing process of warming, what we call the trophication of the Mediterranean, that has very, very negative effects on the Mediterranean biota. And there is also an acidification process that is not shown on that map. But you are absolutely right in your comment.
Okay, thank you, Michele.
But these two really tell you that there are excellent, important socio-economical reasons to shift from shellfish agriculture, mariculture, to algal mariculture. Because while these are affected, all calcifiers are affected negatively, algae are affected positively. Okay, so why go to algae? First of all, we saw that they are more efficient, hundreds of times more efficient than land plants, because their quantum yield is higher, the aerial efficiency that translates into income is higher. Now we go just to have a look at the different farming methods. Here you can see Japanese or Chinese farms of seaweeds that are used, seaweeds, macroalgae, are used for food. For example, in Japan, 60 different seaweed species are eaten. So this is hundreds or maybe thousands years tradition, especially in Japan, as you know, is an island nation, and that was the tradition there. By the way, massive efforts and incentives were invested, especially by the United States, into popularizing and increasing the consumption of meat in Japan and in China, of course, with catastrophic ecological effects. Just a comment. Today, are used to grow our animals for food, primarily cattle, 80%.
Sorry, Zvi, can you repeat the last sentence because we missed the signal? You said that in Japan there are more than 60 different species of algae that are cultured. Can you repeat?
And not only culture, many of them are harvested from the wild.
Okay.
I mean, on the menu, on what they eat, are 60 different species of macroalgae.
Okay.
By the way, that also created for them an acceptance of microalgae. For example, I have seen liquor from microalgae. I've seen green yogurt from macroalgae. I have seen pasta, green pasta from microalgae. So they are used to it, and they immediately moved into adapting it to modern requirements. Here you can see I'm now into showing you some different culture systems. Here you see a culture system for astaxanthin in the south of Israel in tubular reactors. They are freshwater algae, so they cannot be grown in open ponds, because immediately you would get replacement by the algae you want by any other algae, also organisms that eat the algae, so you have to use something of that kind. And here you see the plant in again near Eilat, where they grow the Dunaliella for the beta-carotene content. First of all, on the right, they create situation for rapid increase in the numbers, in the concentration of the algae. Then they are transferred to nutrient-limited situation and to such exposure that they get a lot of light. And then the biosynthetic process is directed toward maximizing the amount of cellular content of beta carotene.
So I want to underline, this is in the middle of the desert, near the Red Sea, and then the fresh water, they come from desalinization.
Stefano, what you see now is also 20 kilometers north of Eilat, in the middle of one of the driest deserts, almost never cloud, extremely high sun radiation. Here, they can use open ponds because the salinity of the medium in which Dunaliella salina is grown is such that no other organisms can live.
Yes, that's from the Dead Sea. Yes, yeah.
Yes, it's high salinity, and also the culture cycle is very slow and very fast. So you harvest in 10 days, in 2 weeks, and there is no time. If there was any infestation by something you don't want, it wouldn't have time because these would grow faster.
So this water comes from the Red Sea, the water.
Yes, the water.
Yes.
But it's even higher salinity than that.
Yes.
And here you can see
also the water in the tubular reactors come from the Red Sea. No, right? It's fresh water.
Fresh water. It has to be desalinated water. Fresh water.
Thanks.
That is the alga here is Hematococcus that we saw before. The freshwater alga that produces astaxanthin, the very, very valuable product.
So I also have a comment on these pictures here, that you see some clouds. Maybe that's the only times in the history that there were clouds, because there's no clouds there almost. It's impossible to see clouds in the south, like in the Negev desert. So this is a very peculiar pictures.
Yes, but still you get there, the average rainfall is about 50 millimeters a year, which is an extremely dry desert. Okay, now you have here a few illustrations of the combination of things. So you can use wind stirring, you put seawater, you put sewage that as a nutrient source, and the result is that the whole system treats water, so now you can discharge the effluent after the algae removed virtually the nutrients, so it doesn't cause eutrophication of the receiving waters, let's say marine coastal waters or rivers or whatever, and that water can be recycled into the culture system. And by such systems, here the algal culture circulates, and you can harvest them in that case for fuel, lipids for biodiesel, and sugars for after fermentation to bioethanol. So the same thing, but a little bit shown in a different way. You can see again CO2 from industrial sources. You can see manure from animal farms. You can see seawater, and that combines into explaining that it is not competing with traditional agriculture. That is not for area, not for water and not for, let's say, for fertilizer, because you derive it from the manure. You arrive it, and at that time you get income from treating wastewater. Also, there are incomes. There is income for absorbing CO2. These are called CO2 credits. Now, all of these are basically financial gains. Now, we showed that if you find a way to create, instead of constant sunlight, fluctuating sunlight, let's say by the algae going up and down in the ponds, you increase significantly the photosynthetic rate and production. Now, one of the main costs of harvesting the algae is the very process, the energy-intensive harvesting. And one of the methods we have been testing is including in the algae, while they grow, magnetic ferritic particles that are, by the way, compatible and allowed in food. And then you can use these algae to extract sugars, as you saw before, to extract pigments, to use for transportation. And you can grow them in such photobioreactors. And we have also played with a layer of algae and the second layer of photovoltaic devices. It works in a very interesting way because, as you know by now, the algae do not harvest the green light, and the green light is the most efficiently used light in photoreactors. So you get here electricity, you get the algal biomass, you treat sewage, wastewater, and so on. And again, you can see algal farms in different ways, in lagoons. And here you can see our laboratory at Bar-Ilan, where on the smaller scale, we can use the changes in medium, starving them for the increase in certain products like pigments. And all of this is computerized, and we can follow the changes in the different culture parameters here, measured as use and evolution of oxygen as we change different mixing regimes by bubbles or by paddles. And here we go into the different phases of the production. You see the cultivation, you do then starve them, as I explained in the case, in the example of Dunaliella, and then you have, this is in Hematococcus, and they are extracted, and here you have the product. Now, in each of these stages, you have economical consideration. You have to decide which of the algae you use, and there are tens of thousands of algae, and only about 10 species are already exploited, which means that there is still a lot of room for improvement. And I'll tell you more than that. I'm pretty certain, because of that, that in 10 or 20 years, different algal species will be used in the culture, species that have not been studied yet. Then we already are working with manipulating the, by molecular biology tools, by enhancing the genes that produce the compounds of interest. Then comes the cultivation system. It has to be sterile or non-sterile. You have to move from the lab scale where you grow the seed cultures, the inoculum, and then go into harvesting. You have flocculation that is done by adding an aluminum compound that combines with the algae and can be afterwards recycled, or you can do flotation. It's by little air bubbles, or centrifugation or filtration, depends on the kind of algae that you use. Then you have to dry it by solar, by spray, by freezing. Again, it depends on the economic value of your final process. If it is expensive, you can use anything. And if we talk about prices like beta-carotene or hematococcus, you can really do whatever you want, because the harvest cost will be very small component in the final product. But if you want to make biodiesel, this will become a major consideration. And again, you can see how you combine solar and wind and CO2 to produce your algae, then refine, and then again the concept of producing biodiesel. By the way, both Exxon and Mobil's among the giants in the fuel industry. Both of them have massive labs of algal culture. And here you have our vision of the same thing. You can see how the algae are grown and the stuff that you can get from the algae. You can feed fish, and that is very important to understand that all the larval stages of shrimps, of fish, and the algae themselves, in the case of oysters, depend on their early larval stages on algae as food. So there is already a large enterprise everywhere where they grow algae, where they grow any marine organisms. It's called usually in the local vernacular, it's called an algal kitchen, where they grow these algae to which they have to feed the larvae of fish, of finfish, of fish, or of shellfish. And then you go to market. Some of these, the markets obviously exist. Some of them, especially in Europe, the markets have to be developed and created. Just see the massive increase in aquaculture of macroalgae, and see that the contribution of the wild harvest is very small, and it's not economic, because these stocks are readily depleted, and then you are left with nothing to harvest. Here you can see the European stage for the culture of macroalgae and of microalgae. You can see that Italy is not very active, and spirulina, that is strictly speaking not an alga, but it has a massive market in food additives, is already cultured a lot in the south of France. And you can see from that there is a lot of room and opportunity to increase both fields of macro and microalgal culture in Italy, that where it's compared to other countries in Europe, it is almost non-existing. But you can also see the projected dramatic increase in all the applications of algal culture in the world. Europe and others, you can see that. And here you can see also the global distribution of the culture of algae. You can see that in China and the Far East, it's highly, much more developed than in the rest of the world. Now, one of the most interesting future venues of mariculture is polyculture. It's where you grow a number of species. In this cartoon, fish are grown, and in that culture, shrimp is the same. They pollute the water, but that pollution is exactly what the algae needs, because that provides the nutrients for algal growth and multiplication. And then the algae can be fed to oysters. That's the idea, the very simplified idea of polyculture. And really the result is that the mariculture is prevented from polluting the environment. It's a major problem. All the fish ponds and the fish ponds and the oyster ponds in Asia and other countries pollute the environment and pollute the marine environment to an incredible degree. And here you can see again a combination of culturing many species with macroalgae, with phytoplankton, and you see the recycling of nutrients that is needed for this. You can see the cultures of oysters, you can see the phytoplankton, and you can see the culture of macrophytes, of macroalgae. And you can see another cartoon basically of the same idea. And really I would say that in the open marine systems, marine lagoons, probably the way to go is polyculture. And here you can see examples of polyculture. When you grow algae, you grow urchins, you grow shrimps. And here you see again the same. The ulva is cultured basically on the wastewater coming from fish, and in this example of urchins. And what is not shown is that the ulva is the main food for growing abalone that sells in Japan for incredibly high prices, for tens of euro per kilogram on the shell. These are marine diatoms that are phytoplankton algae that are still not exploited. They are enclosed in little, really glass containers, and that glass is very, very useful in different industries as an abrasive, very mild abrasive, even in toothpaste, and also in science in chromatographic columns. These are other species. By the way, I took these photographs in the Antarctic. They are dominant phytoplankton species with great economic promise. The surface, the warming of the oceans is also a very important factor because, as I explained before, they increase dramatically the respiration losses in any algal culture. And here it only shows that as temperature goes lower, you can get a doubling of the algal population in much, much less time. So if the water temperature is -4, you get algal doubling already in 2 days. But if you move to 5 degrees centigrade, you need like 10 days. So that shows you the adverse impact of the warming of the Mediterranean on your gains when you grow algae. You pay more for respiration costs. And this is something from our laboratory showing the dramatic increase if you chop the light, if you expose the algae to different frequencies of fluctuating light. Here we have a cartoon produced by our postdoctoral colleague, Dr. Saeed Abu Ghosh in our laboratory, that analyzes where are the costs and where you should and could increase the financial gains from culturing the algae. And that is the end of my presentation. And if you have any questions, I shall try to answer them. If I tell you that you have a very good question, means it's so good that I don't know the answer.
I have a few questions. Can you go back to the 3 slides when you show the flashing light? This one. This is the flashing light effects on.
Yes.
Can you explain a bit this concept?
Okay. I'll explain it a bit in a more complicated way. First of all, if you want to grow algae, you are going to grow them on high light, because then they grow faster. But what happens when they are exposed to high light, they start losing on respiration, and there is also an investment in the xanthophyll cycle, and there is an increase in respiration that is used just to correct the damages caused by the high light intensity. Now, if you give high light for a very short period, let's make it simple, for 10 seconds, and then instead of continuing forever, you intercept a period of darkness. So all the high light enhanced respiration and the operation and generation of the xanthophyll cycle are long processes that take minutes. So if you interrupt the exposure to high light soon enough, none of these losses will take place. So you create a period of darkness and turn on the light again. But you switch off the light before the losses start. In this, we only look at one effect. What is the effect of light frequency? And you see now that at higher frequencies of the flashing light, you have an increased rate of photosynthesis. Now, you get into economics. How do you create flashing light? So if your product is expensive, you use light-emitting diodes or whatever. But if you are into something less expensive or totally inexpensive like biodiesel, then you must use sunlight. So you must find a system where the algae are exposed to light and dark cycles. And that can be accomplished by designing, by the engineering design of the culture systems, so that while the algae flow, they move from a high light to a low light place or segment of the pond or segment of the tubular reactor, and that creates the fluctuating light. And in the works of Dr. Abu Ghosh and Dr. Iluz, my colleagues at the lab, they got 2.5 times with fluctuating light than with the constant light of the same photon dose. You have to compare, and even higher, we found that the highest we get, not when you get light dark, but when you get light, very low light, very low light. So we use two terms, flashing light if it is light and dark, fluctuating light if you have high light and low light. And the low light is not flashing, it's constant. And that prevents a process that is negative, and that is the loss of states, of S states. There are 4 states in the emission of oxygen. If you start, you need 4 electrons moved. And if you keep long enough in the dark, it reverses one of 2 phases. So if you keep a very low background light, you avoid all the losses of energy that I described before, and you prevent the decay, the spontaneous decay of the S states. Now, all of the things that I try to bring over are ecological, economic, and biophysical considerations that you must take into account if you're planning to become a millionaire by growing algae.
Zvi, but the company that are now actually have in place mariculture, algal mariculture in Israel or in other country are aware of this increasing productivity with flashing or modulating light. They are already like using this
No.
discover, or just is only.
Now I'll answer. You see, in the natural environment, in the sea, and we learned from that, when you have a wave, it acts like a lens. So if you have macroalgae growing on a surface at 2, 3, 4 meters deep, because of the lens effect of waves, they see already fluctuating light. Because, and if you swim, Stefano, over a sandy bottom, you can see these lines of light.
Yes.
Okay, the intensity of this for a tenth of a second can be 10 times full radiation of the sun. So shallow water organisms, I don't want to explain, but that phenomenon lasts only in the upper, because then the focal length of the waves is such that these lines cross and you wouldn't see them at, let's say, at 20 or 30 meters deep. Okay, so that already takes place. We don't know how much, if and how much it does contribute. The fact that an alga seated at 2 meters deep sees fluctuating light. And because the waves, you don't see dark. It's not zero. So you see high light, low light. Then in the ponds, if the mixing is the alga will spend part of the time near the surface, part of the time at the bottom. So in this strip, the alga already sees a fluctuating line. But it has not been studied in the way how to mix, what depth to mix, what kind of device you use to mix, paddles or jets or whatever. So this, all that field is very, very new. And it's really publications that came out in the last 2-3 years. So this is another field. And again, the question is always, what is your product? Is it expensive or not? For example, for culturing chlorella for the green, I don't know, for the green ice cream, which they make for the green pasta and all of that, the price is so high that they grow it in dark ponds illuminated by artificial light. So the cost of electricity is not even a component in the final cost of the product. So you always have to keep in mind what is your target product.
So are there any questions, comments from the hall, from the students?
Yes, I have a question.
Grazie, Alberto.
During the presentation, you showed that if the intensity of light increases, the content of polyunsaturated fatty acids decreases. And so I was wondering, is it for an anti-oxidative action, or for which reason is it so?
Okay. First of all, we were surprised by that finding, because we thought initially, intuitively, that the need for antioxidants will increase under high light because of the formation of free radicals. It turns out that it is the opposite, at least in the few algae that we studied. And that connects immediately to the PUFA question, because there are components in the thylakoid membranes. So as you get 10 times, 20 times more of these membranes, you get more of the polyunsaturated fatty acids, and you need the antioxidants to protect them, because they are extreme targets for the action of oxygen and free radicals.
Okay, thanks.
Zvi, what is the GDP, the mariculture economy in Israel now, in terms of GDP or people employment?
Frankly, I don't know. There are the two microalgal ventures near Eilat, and there is one macroalgal on the Mediterranean. But I think that today most of the income of algal culture comes from the two ventures near Eilat. And they don't tell so much about the income, and at least I didn't get the data to present them here.
Okay. Are there any other questions, comments from people online? There are a bunch of people still.
Yes, I hope in a couple of years to be invited by Stefano to visit your successful algal growth systems all over Italy. Okay, Stefano?
Yes. So, by the way, one of the next fellowship will be on this PhD program will be on polyculture. So let's see, maybe we'll have a student enroll in this topic.
Yeah, I think that for ecological reasons, the interest and the investment in algal mariculture and in algal culture in general has to increase by any economical model is going to increase. As you saw already, the projections, all models agree that it will have to increase in the coming years. By the way, from a totally political angle, that the replacement of Donald Trump by Biden is an important global turning point for all the sustainable economic ventures and also for the future of the planet.
Okay, so if there are no comments or question, I would like to thank you, Svi, for the lesson. We recorded this lesson. I know Fiorella started the recording, so I remind her to also to stop the recording when everything is done.
Stefano, I can offer something. If you want, you can circulate the PowerPoint presentation.
Yes, in fact, that was my question. Next, if you can send me today a PDF of the presentation, we will post it together with the record, with the video in the website of the program, of the PhD program. So yes, please send me the PDF.
It's not a PDF.
Whatever, a PowerPoint, it's okay.
Because it's a huge file, I'll have to send it by special way, but I know how to do it.
Yes, you can use WeTransfer, for example.
One of these, yes.
Or something.
Yeah, I should do that.
Okay, so this was the last lesson for this 2021 teaching week of our PhD program. I guess was a successful week, full of interesting topics, and I thanks all the students for being present and on time and all the lesson. And now it starts season for you student to start also putting together the thesis, at least for the oldest students, the 34 cycle students, and we will get in touch soon for other related matter. So
yes, and I would be very, very glad to host one of you for your PhD research.
Oh yes, I actually was really impressed by the slide of your lab. If you want to put it back 1 second, the Bar-Ilan facilities, it was quite impressive. Where is? Yes, this one. This is your lab, just the lab you have only for your project on algal photosynthesis, algal biotechnology.
Yes, we also have the aquarium system that was established for the, for the coral worm.
Yes, yes.
It's in addition to that, but it can be used for algae.
Yes. Okay, Svi, thank you again. We'll keep in touch and have a nice